My name is Eric Mörth, and today I want to present you Ask and Reveal Natural Language-Driven Analysis of Multiplexed Immunofluorescence Imaging. With this work, we want to answer the question, can we use LLMs to help exploring highly multiplexed microscopy data? What you can see here is an example of CISIF imaged tissue from a skin cancer patient. In this example, you can see 8 of 54 channels that present proteins within the tissue. The proteins tell you about functions of individual cells that you can spot here in this image. And furthermore, we can also see larger structures like in yellow collagen fibers. Within a certain area of this skin, we can see cancer cells in green, and then we can also see immune reactions in turquoise and in violet. What makes this data challenging is first its size. It can go up over like 1 terabyte of data of a large scale of a tissue. And furthermore, we have a lot of channels, in this case 54, but it can also go up to over 100. And this makes it especially challenging to flip through all of them and find interesting areas in the tissue that answer certain hypotheses. So how are people typically looking at this data? They can use tools like Vitesse that was developed in the HiDive lab. And you can see here on the left you have your tissue image, and on the right you see the individual channels. And you can toggle through them individually, and you can select them here out of this drop down, and then you give them a color and you change the thresholds, and then you can see the final image that you then start to inspect. But this is very cumbersome and takes a lot of time and takes away from actually analyzing the data. So what we propose is introducing an LLM agent into the same interface that you can speak with or write with and explain what you actually want to see and explore. So what you can say here, show me tumor in my T cells. Then the agent can translate that into the individual protein channels you're interested in, and then change the configuration of Vitesse to actually show you the result you're looking for. Furthermore, the agent can also explain you for each individual protein channel what they mean and what they stand for. So how does it now change the way we work with MIF data? So first, it's of course convenient. And second, it's also easier for onboarding, because biologists are primarily interested in finding areas within the tissue that confirm the hypothesis or interesting areas to find new hypotheses. And so they don't really want to learn new tools, but they want to just use them. But more interesting is also, can this also help us finding regions of interest? Because as the tissues grow larger and we don't see all the channels at the same time, it would be very beneficial that there is a support to tell me where to look. So we looked into this, and the first idea was to give the image to the LLM and let it find the areas himself, but this is not possible because you have so many channels that you can't even create the image. So we looked into laying a grid on top of the data, and then we can explain the data for each grid cell based on statistics, and then ask the LLM to find brick cells of interest. So we went in, laid this grid on top of our data. As you can see here, this is the marked 1 channel, which stands for tumor. And then when you lay the grid on top, you can see like a more blurred image of the same data, and you can find some highlighted spots. And this data can actually be queried by the LLM and then can be combined with multiple channels to tell us where there's interesting regions to look at. So how does it work? In this grid, for every channel we get a statistic value like mean, average, or also like standard deviation of the protein channel distribution within that brick cell. And then the LLM can translate questions like show me this and that marker into like finding which IDs those markers have, and then also defining how many of those bricks are of interest to be retrieved back. And then finally it calls a function to retrieve them and then shows them to us. So in the actual interface, we can now say, show me areas where there are densely populated T cells, for example, translates that into the channels that mark for T cells, and then finds us the region where those are like particularly active, which you can see here in blue. And these are then the areas where the biologist should have a closer look and then see what's going on there. Then we also asked the question, can the LLM also give us insights into tissue processes visible in the image? So the other way around. And that also works, like you can mark an area in your tissue, and then we retrieve those bricks and give the statistics of the channels you're currently looking at to ChatGPT, and it can then start to reason and argue what's going on here. And in this area, for example, it just knew the channel markers and the distribution, and it can tell us that there's like a melanoma presence, and there's also some immune reaction going on just based on those 4 markers. Furthermore, you can also ask what's going on throughout all the markers you have available, so even stuff that I don't even see, and it can argue about these as well, and then tell us that there might be some different markers we also want to look at because they're also active in that area. So how does it work? We're using multiple agents that work together. So one is a supervisor agent that can then hand off some tasks to the MIF expert agent. So this is the one that reasons about the biology in the tissue. That can also be something that is purely based on recent papers. Furthermore, we have an SQL agent. So that's the agent that translates region-based queries into finding the bricks of interest. And finally, we have an agent that actually writes the test configuration and updates our user interface so we can actually see what the results were that were given back by the other agents. And so the supervisor decides based on the question of the user which agent to call and how to then further proceed with the results it gets back. So what are our next steps? We want to explore more data sets with this approach by giving it out to biological experts, and they can just use their own data sets in the same way. Furthermore, we want to improve the 3D capabilities, so we can do the search both in 2D and in 3D and retrieve back bricks. And then we want to validate the reasoning results together with biological experts. And finally, we also want to validate with different models besides ChatGPT. With that, I want to invite you to also check out our poster because there's more examples there and a more in-depth discussion. If you have any questions, don't hesitate to reach out to me or my co-authors. Thank you.